Okay, good morning. I'm Jason. I'm a postdoc in Ed Butler's lab at Cornell University. I'd like to thank the organizers for inviting me out, and we're going to walk through some case studies that I've been working on with collaborators here and elsewhere on applying genomics to crops for the developing world. So first, let's take a step back. Let's look at the motivation. This is the motivation for why I'm in this field, and I suspect a lot of you are too. It's global food security. There are different definitions floating around, but one most people can agree on is that food security means you have enough food, both in quality and quantity, to lead a healthy and active life. About 850 million people right now are estimated to be food insecure, and increasing food security has been shown to be one of the best ways of increasing not just nutrition, but also educational attainment and even political stability. So this is a good goal. But there's lots of stuff that's impeding us from getting there, some constraints we have to face. People have already mentioned most of these. Increasing population, more mouths to feed. Changing consumption habits. The world is generally getting richer. Diets are diversifying, which nutritionally is generally a good thing, but it means that more resources are required to feed the same number of people. Environmental variability is going up, climate change and other factors. And while addressing all of these, we have to do it without completely trashing the natural environment in the process. Agriculture is arguably the biggest impact humans have had on the natural environment. And as we move forward, we have to keep that in mind as we try to solve these other problems. Now, this is a really big problem, and I don't claim to have all the answers to this. Trying to achieve that involves work from all sorts of different sectors. Crop breeding, agronomy, government, infrastructure. We could spend an entire conference just going over all of these, and I'm not going to do that. I'm going to focus on my little portion, which is kind of what we're focusing on here in this conference, which is the plant breeding, the actual improvement of crops. And when you get down to it, crop improvement is mostly crop genetics, because an improved crop is one whose genes are better suited for the environment in which it finds itself. Now, we're currently in the golden age through what's been shown already. You've already seen this graph several times. The cost of sequencing. Around 2008 or so, the bottom really fell out of the price of gene sequencing. And suddenly it became not free, but a lot cheaper to get genetic data. And this has opened up reams of data. This is completed plant genomes. It's a little dated. This goes up through 2013, and I think the Africa Orphan Crops Consortium has already more than doubled the number on here. So we're getting enormous amounts of genetic data. Beyond this is just the transcriptomics, genetics, genotyping, all sorts of stuff. This lets us open up hypotheses and test questions that we never would have been able to even approach 10 years ago. And so I'm going to walk through some case studies on different ways these can be applied. These are collaborations I've been doing either here at ICRISAT or with CIMMYT, and we're going to pretty much go up the resource chain. So starting with Barnyard Millet, which is a very neglected crop, then up through pearl millet, which is technically neglected but is among the least neglected of them, and then maize, which is arguably one of the best resourced crops in the world, and just showing how these approaches can work at any scale. So first, Barnyard Millet. Barnyard Millet is one of ICRISAT's mandate crops. It's a minor millet, two species actually, Echinocloa colonna from India and Echinocloa crusgali from Japan. They're alternative crops here in southern and eastern Asia. They're crops between the major crops, or in case the major crop fails. In the US, we grow it as forage, and some clever marketer gave it the title billion-dollar grass. I have not been able to track down who did it, but I hope they earned an award from their company. And with this, the goal in this project was to characterize the core collection that had just been created here at ICRISAT of about 90 Barnyard Millet lines. There was essentially no genetic data on this, and so the goal is just to see what it is, get a snapshot of diversity. So my tool in this was GBS. This has been mentioned many times. I'm not going to belabor the technique, but I do want to go over the pros and cons of it. The advantage of it is you get your SNPs in the same step that you find them, so you don't have to discover them and then go out and genotype. It's a simple protocol. You don't need a reference genome, although it really helps. But the one most people like it for is you get lots of SNPs pretty cheaply, and it seems to be pretty broadly applicable. But again, no platform is perfect. Sequencing errors do look like SNPs. This is an issue with any sequencing-based platform. There are ways around it with filtering and such, but you will have to deal with it. And the big one is missing data, because You're doing sequencing, it's a stochastic draw. So you get uneven coverage. And again, there's ways of dealing it with depth and with filtering, but it's always something you're going to have to deal with. Overall, I found that the advantages outweigh the disadvantages, but we've already seen plenty of platforms, and so there's other choices that could have been made too. Now, when I first ran the SNP calling pipeline on Barnyard Millet, we had 95 samples. I got 20,000 SNPs off the pipeline, which is great. But there's one problem, because both Barnyard Millet species are hexaploid. And this, as was mentioned yesterday, when you've got polyploid genomes, you get alignments across the genomes that look like SNPs, when in fact those DNA segments have no business being aligned against each other. And you can see this in the site frequency spectrum. The ideal one, based off of population genetics, looks like this. You've got lots of rare SNPs and a few common ones. But right off the pipeline, this is what the frequency spectrum looks like. I've got the two species in red and blue, and then the combined one in green. It looks nothing like what it's supposed to. So most of the SNPs in each species end up being common, supposedly, and then you've got these three little humps. Well, all these ones over here aren't actually SNPs, they're paralogs. They're lining up across the different genomes in the plants. And then these three humps are alleles that are segregating differently in the two species. They're fixed in one and still segregating in the other, or something like that. But basically, this is a bunch of artifacts, and if you're trying to do any population genetics on this, this will break just about every assumption in the model. So, thankfully, we know that these lines should be almost entirely inbred. And so by simply filtering by heterozygosity, we can suddenly get back actual useful SNPs that meet the assumptions of the PopGen models, and then we can then use to do diversity analysis. You'll see that in the combined one, there's still a little hump there. Those are the SNPs that are fixed in the two species, but fixed differently. So these are the SNPs actually that you would want to use to discriminate which species a certain plant belongs to. So then taking those SNPs, we can do some basic PopGen analysis. This is phylogenetics, just looking at relationships among them. And the first thing that comes out is that, as expected, we can clearly separate the two species of Barnyard Millet. We do end up with some things here in the middle that look like hybrids. It is known that these can hybridize in the field. Those hybrids are supposed to be sterile. So we're not entirely sure of the source of this, but we are expecting it's probably some sort of artifact, either pollen contamination or something like that. Then when we zoom in on each species, we get some very nice population clusters that separate out. And this is especially nice because there are existing classification structures into races and subraces, but it's known that these don't actually correlate with their point of origin or even their genetic relatedness. They're just based off of morphology. And these we know have some nice characteristics, these genetics-based ones. So if you actually plot them on a map, they segregate geographically. We don't have passport data for many of the accessions, but for the ones we do, you can see when you plot them on the map of India, you get a very nice geographic distribution. We do not have any passport data for the Cruz-Gali accessions, but they're the ones from Japan. So I strongly suspect that these different clusters are related to the islands. I have no hard data to back that up though, so that's just speculation at this point. And so from this, we now have a snapshot. It's a way of starting to choose lines if you want to build populations. It's starting to see if you have underrepresented diversity, and it's a building point. First pass. Very basic, but still a good place to start from. Now let's move on up the ladder to pearl millet. So pearl millet is the least neglected of the millets. There's actually quite a bit of resources devoted to it, especially here in India. It's a staple crop in sub-Saharan Africa and India, especially in the Sahel region of Africa, very important. The genome is pretty large, 2.3 gigabases, so nothing compared to wheat, but it's on about the same order as maize. And the reference genome is in process. In fact, this is actually what I'm going to talk about as part of making the reference genome. Most of the sequencing work was done by BGI, and they assembled the contigs and the scaffolds. My job in this was then to take those scaffolds and stitch them into pseudochromosomes. Because those of you who've worked with genome assembly, you know that just due to technical limitations, you can stitch the contigs up to a point, but you can't get a whole chromosome together without some outside information. And so I used several mapping populations I had on hand to create genetic maps and then to build the pseudochromosomes together. I had 3 populations, one from ICRISAT here in Patancheru, one from ICRISAT in Niger, and then one from a collaborator in Georgia in the southeastern United States. And each of these populations had different strengths and weaknesses. So, for example, the Tifton population from Georgia had deeper genotyping depth than the other two. So, much more confidence in the genotyping calls, much better coverage, less missing data. But it also had many fewer SNPs. And that's because it was a cross between elite US lines, the narrow germplasm. And in fact, as we went through this analysis, we realized there was a large chunk on one chromosome that had no SNPs because it inherited the same haplotype from both parents because they shared a common ancestor. So, different strengths and weaknesses, but by combining the different populations, we can get a decent map order out. And this is the way that we did that. Excuse me. So, this is what the SNPs look like right off the SNP calling platform. This is a linkages equivalent plot. The SNPs are down the y-axis and across the x, and every point is an intersection between them, a relationship. So a SNP with itself is down the diagonal. This upper triangle has the strength of the relationship, the R squared, a measure of LD, and then the bottom is the statistical significance. And right off the SNP calling pipeline, they're basically random. Because they're small contigs, there's not much relationship there. You might find a few small clusters, but essentially they're random. So the first step is you hierarchically cluster them to get linkage groups. We start seeing most of them here, these blocks along the diagonal. These off-diagonal blocks indicate where linkage group has been split. So then it's time to repair those up, and now we have Cornell 7 linkage groups. And this is that small one I mentioned that has fewer SNPs because in this population there's a large chunk of identity by descent, IBD, inherited from both parents. After this, it's just ordering. I use MSTmap, common software off the shelf, to do that. And finally, clean up in RQTL, which I know it doesn't look like the map change. It did, but it was very small-scale stuff. So after doing this for each population individually, then I merged the maps using using merge map and then also some custom software. And in the end, we end up with a pretty decent reference genome. Got almost 5,000 contigs ordered, which is a minority of the contigs, but it's 93% of the sequence data. Most of the remaining contigs are very small. Many of them don't even have any SNPs on them. So this was a pretty good coverage, actually. 60% of the contigs we even have putative orientations for. I will say right now, there are going to be mistakes in this genome. As the person who put it together, I can guarantee it because there's limitations on the data. But it's a pretty good assembly. It's much better than what we had before. And just based on my own experience, having any sort of genomic data, even if it's just scattered across 100,000 contigs, gives you a lot more power for detecting variation and doing genomic work. So this is assembled now. The manuscript is in preparation. If you want to know more about it, talk to Rajiv, because he's the one in charge of that. And now we move on to the last case study, maize. Trait mapping. This is the ultimate goal of all of it, isn't it? It's to find genes that are controlling certain traits and move those into useful germplasm so that it can be deployed to help smallholder farmers. So this is a collaboration I've been doing with CIMMYT on their water-efficient maize for Africa. These WEMA families are 20 biparental families, each of which has about 200 lines. And the goal, well, I should explain. The project, as originally conceived, was to generate these lines and use them to do QTL mapping. But the idea was just to map each of them separately as a separate biparental family. But now that technology and methodology has progressed, CIMMYT really wanted to combine information across the different populations. It's sort of similar to how you work with a NAM population, except that there's no common founder here. It's just 20 different families that you try to combine data for. And so that was my challenge, is to try to develop the methodology to get this to work. Now, it's a little tricky because since these were not actually designed to be analyzed together, they were never grown all in the same environment. In fact, no environment ever contains more than half of the families at any given time. And they're different halves, so they don't actually overlap except for 2 check lines, which makes it a little tricky. Possible, but tricky. So I took 2 parallel approaches to this. One is just traditional joint GWAS. This is the one that takes advantage of those check lines to create BLUPs, best linear unbiased predictors, across all the environments and map off of those. This is sort of the tried and tested way, and it was something to compare against. The other way, and the one that I find a bit more exciting, is a Bayesian GWAS approach. Bayesian Statistics is just a framework that lets you deal with probabilities and updating probabilities. And the important part about this is that it's modular. I mentioned that nothing was ever grown in the same, that all the populations were never grown in the same environment. Well, it's actually more than that. It's that I can track down that 4 or 5 of these families were grown at the same field station at the same time. Their physical relationship on that field station is completely unknown. So for all I know, they were grown kilometers apart in entirely different fields. It makes it very difficult to assume that they're actually in the same environment. So what this lets me do is I can actually map in each environment separately, each field population combination, and then simply combine the probabilities at the end in a very quick, very easy analysis. It makes it very modular. And so I think this has some good potential. It's sort of proof of concept right now to see if it can work, but so far it seems to be working pretty well. This is a comparison of those two, looking at days to flowering. This is, again, flowering time is something we already know the answers for. It's a good way of checking if our methods are working. The traditional GWAS up here shows peaks. We get some nice resolution here. Chromosome 8 has all sorts of stuff everywhere across it because it's where a lot of flowering time genes sit. And then the Bayesian GWAS down here at the bottom, you can see has very similar spread. It's not exactly the same, but it's pretty close and it indicates it's working pretty well. If we zoom in on that chromosome 8, you can see we're even hitting some of the known genes, ZCN8, VGT1. These are well-established flowering time genes. One nice thing is we're also hitting these peaks that have no known candidate genes near them, which is nice because these candidate genes were generally discovered in temperate U.S. germplasm. And the WEMA populations are tropical germplasm, which means we might be finding some genes that have just been missed before because we've been looking at the wrong materials. So this is still a work in progress. It's not completely done yet, although I hope to be wrapping it up in the next few months. So there we've got 3 case studies. And the take-home message from this is that you can take these genomic technologies and apply them to any level of crop breeding. They develop tools to help breed things faster and better. And, I mean, to be honest, one take-home from this is that the genotyping problem is basically solved. I mean, maybe the different company reps here will disagree, but the challenge here, the question is no longer, can I get genotypes? It's which platform is best, or which gives me the best value for my cost. But if you want genotypes on your material, no matter what it is, you can probably get them. So genotyping is no longer an issue. Going forward, I think we have different challenges we need to meet. The first, as has been mentioned, is phenotyping. I like to say that genotyping is not just dirt cheap, it is actually cheaper than dirt. It is often cheaper to get a genotype on a line than grow it out in the field and measure its phenotype. And so we need to figure out a way of getting the cost of phenotyping down. There's work along these lines. Our lab uses rapid phenotyping with barcode scanners, but high throughput phenotyping, where you drive a tractor through the field or just take a snapshot from a drone, this is really where we need to work. ICRISAT is already doing a bit of this. You may have seen their little phenostat out there, where it's taking measurements on like 2,500 sessions every 2 hours. Great progress. We need to figure out a way of deploying this out in the field, any field, so we can get this level of data. Which leads us to the next problem, data infrastructure. We're already drowning in genetic data. Pretty soon, we're going to be drowning in phenotype data. And so we need to develop the tools to actually deal with this and make it so that the data is useful. This means investment into servers, into databases, common exchange formats. It'll be a big problem, but it's one we have to solve if we actually want to get this going so we can take all this data and translate it quickly into usable breeding outcomes. And last one, faster breeding methods. This is a schematic for genomic selection, which has already been mentioned, and I think we're going to have several good talks on that later in this conference, so I'm not going to deal a lot with that. But the basic idea is you speed up breeding, in this case by being able to skip the phenotyping step a few times. Other options are also out there. Site-specific genome editing through CRISPR-Cas or Talons or whatever your platform of choice is. Ways where you can move an allele directly into your elite germplasm without having to worry about linkage drag. Other things that maybe we haven't come up with yet. But we need to breed faster because in all of this, time is our most limited resource. We don't have the time to wait for solutions to just come about slowly and arguably naturally. We need to push it faster in order to address all these challenges that are coming up. So that's my take on these issues. Now, obviously, a lot of work has gone into this, and it wasn't just me. I've had very good collaborators here at ICRISAT and elsewhere, chiefly Tom Hash, Rajiv Varshney, and then Shuai Zhang at Mexico. Also a lot of support back at Cornell, and of course the various funding agencies that have made all this research possible. And so thank you very much for hearing my 2 cents. I'd be happy to answer any questions.
Thank you, Jason. Again, sparing us some time. We can conclude the session in well before the projected time. We can take 3 questions if they are.
Oh, come on, all of that and no questions? Ah, there we go.
So no question?
Can you select for the homozygotes in segregating F2 population?
By that, you mean if you genotype it, can you look at the ones that are homozygous in a certain region and select those? Yeah, as long as you have the genetic information, you can just look for which ones are homozygous in your region of interest, yes.
Yeah, because that would certainly reduce the breeding time.
Okay.
Yeah.
Yes, right there.
Okay, I think it's fine. Thank you. Since you asked the question, so I would like to ask a few questions. So, in nice openly, in your open talk, when you opened the talk, you are very interested about the global security, food security, right? So, and then you went with the presentation, but I couldn't find the clear view of your goal. Of course, you have started with the genomics part and the genotyping, but I couldn't find your all 3 crops. What would be your goal?
Okay, you're going to have to repeat that. I'm having trouble making
Okay,
it out.
direct question. Like, I got your idea of research ideas or objective to solve the global food security,
Okay.
right?
Uh-huh.
So in your presentation, you mainly touched about the genomics part, but I couldn't find the clear goal or view about what you want to achieve.
Oh, about what I want to achieve? Like what my specific goals are?
Yeah, for these 3 crops.
Oh, for those 3 crops. Okay, yeah, I should have mentioned that with the Lima one. So doing, at least in the Lima and the maize, it's doing proof of concept right now to see if the methods can work. Once that's done, then they're going to be extended to actual drought tolerance and water use efficiency, which is the target traits, the reason they were originally made. We're keeping with a flowering time right now because we know the answers, we can check it. For the other ones, it's mostly development of tools. I'll be honest, I'm a methods guy. I let my collaborators pick what the final goal is, and my job is to make it easier to get there. And so I'm not a breeder. I work with breeders, and so my goal in all of this is just help them do their job better and faster.
Thank you. All the best.
Yes, back there. Yeah.
Hello. You detected about a lot of SNPs. How many of these SNPs were actually linked to any genes?
How many of the SNPs are in genes, you said?
Yeah, linked to genes.
Most of the, because of the restriction enzyme we use APK1. It's a common one in grasses. It's methyl sensitive, so it tends to show up in genic regions. Because even if you have a restriction site near a centromere or whatever, it usually tends to be methylated, and so it won't cut, and so you don't see that with this specific GBS protocol. So most of these SNPs are going to be relatively close to genes. Thank you.